In this video, we will learn the principle of media you have used and what the positive reactions should look like. Perhaps this is the most challenging lesson for the Salmonella module. Before we start, let's review the general procedure. On day 1, you conducted pre-enrichment using BPW. On day 2, you did selective enrichment in RV and TT. You also did rapid detection on the same day. On day 3, you streaked the selective enrichment onto XLD and BS agar for isolation. Once you got the single colony on day 4, you streaked them onto TSA to prepare for the confirmation tests. On day 5, for identification, you used the TSI and LIA slant agar for biochemical tests and Salmonella O anti-serum poly A2I and VI for serological tests. Now, let's learn the media one by one. Buffered peptone water is a non-selective medium. Peptone provides nitrogen source. Sodium chloride is here to maintain osmotic pressure. Disodium phosphate and monopotassium phosphate forms a buffer system. The buffer system protects Salmonella from drastic pH change due to the acid production from bacteria or food itself. TT stands for tetrathionate broth. It is a selective medium. You will prepare the TT base ahead of time and add iodine potassium iodide stock and brilliant green stock on day of use. Iodine is the active compound. However, this compound does not dissolve in water well, and potassium iodide can increase the solubility of iodine. Brilliant green and biosalt select against non-Enterobacteriaceae. How to select Salmonella against other genus in the family of Enterobacteriaceae? The iodine and sodium thiosulfate in the medium react, producing tetrathionate. Tetrathionate is toxic to many bacteria but not to Salmonella. That is because Salmonella has tetrathionate reductase. This enzyme could catalyze tetrathionate back to sodium thiosulfate. However, this redox enzymatic reaction produces a lot of protons. The calcium carbonate neutralizes the protons so that the solution will not become too acidic for Salmonella to live. The left tube in the picture is the negative control. The white precipitation at the bottom is the calcium carbonate. When inoculated with Salmonella, as shown by the right tube, TT becomes lighter green and turbid. If you look closely, you will also notice the reducing amount of the white precipitation. That is because when calcium carbonate reacts with acid, they become soluble compounds. RV stands for Rappaport Vasiliadase soy broth. It is a selective medium. Magnesium chloride increases the osmotic pressure of the medium and acts synergistically with malachite green to inhibit Enterobacteriaceae other than Salmonella. RV has relatively lower pH, which is 5.2, and it is incubated at higher temperature, which is 42 degrees Celsius. Those two characteristics also help to select for Salmonella. The left tube in the picture is the negative control, while the tube on the right is inoculated with Salmonella. As you can see, the RV broth inoculated with Salmonella becomes lighter blue and turbid. Sometimes, when the growth is minimal, it is hard to observe any color change. However, if you compare the negative control to your tube against the light, you will see some turbidity indicating the growth. In the rapid detection, you use the Reveal 2.0 Salmonella test. It is an immunoassay. This means the device detects the antigen presented on the bacterial surface. Salmonella have 3 types of antigen: K antigen or VI antigen, which is on the capsule, O antigen or somatic antigen, which is on the cell wall, and H antigen, which is on the flagella. The Reveal 2.0 Salmonella test detects the most common O antigen, A2H, of Salmonella enterica, which is the most common subspecies in Salmonella associated to foods. The reagent zone of the device contains specific anti-Salmonella antibodies conjugated to colloidal gold particles. Upon the presence of Salmonella, the antigen on Salmonella will be bind to the gold conjugated antibodies at the reagent zone. This antibody-antigen complex then leaves the reagent zone and travels to another zone that contains anti-Salmonella antibodies. Those antibodies are fixed and ready to capture the immune complex with gold conjugate. The gold conjugates aggregate in this zone, forming a visible line. The gold conjugates that do not bind with Salmonella antigen will continue to migrate to the end of the device where they're deposited into a waste reservoir. The reagent zone also contains gold conjugate of a proprietary antigen which can migrate when soaked in liquid regardless the presence of Salmonella antigen. Those gold conjugated proprietary antigens migrate to the negative control capture zone that contains antibody specific to the proprietary antigen and are captured and aggregated there to display another visible line. Regardless of the presence or absence of the Salmonella antigen, this line will form when the device is used. This ensures that the test is working properly. As shown at the top of the picture, the positive reaction should show 2 bands, while the negative reaction should only show 1 band. XLD stands for xylose-lysine desoxycholate. This is a selective and differential medium. The selective agent is sodium desoxycholate, which inhibits non-enterics. The differential reactions are complicated. The medium contains 3 different kinds of sugar as energy source: xylose, lactose, and sucrose. Notice that xylose is in lower amount compared to the other 2 sugars. The medium also contains lysine as nitrogen source. The amount of lysine is lower than sucrose and lactose, but higher than xylose. The proportion of each nutrient in this medium is very important for differentiate Salmonella from other microorganisms such as coliform. Carb is the primary energy source, so Salmonella utilizes sugar before lysine. Among the 3 sugar, Salmonella could only ferment xylose. This process produces acids that turn the medium into acidic. Thus, the pH indicator in this medium, phenol red, is changed from red, which is the color at neutral pH, to yellow. However, keep in mind, xylose is in low quantity. The amount of acid being produced in this process is very limited. Once Salmonella consumes all the xylose, it will start to use L-lysine. Salmonella possesses lysine decarboxylase, which could change lysine to an alkali molecule. The amount of alkali produced by this process overwhelms the acids generated through xylose fermentation, turning the medium to higher pH. Thus, the medium becomes pink-reddish color. Salmonella has desulfidrase that could reduce sodium thiosulfate into hydrogen sulfide. Hydrogen sulfide could further react with ferric ammonium citrate in the medium to produce black precipitation, ferrous sulfide. As shown in the picture, the uninoculated XLD is red. A typical colony of Salmonella on XLD is black. If you look closer, around the black center, there is a red halo. BS stands for bismuth sulfide. It is a selective and differential medium. The selective agent is brilliant green, which inhibits non-Enterobacteriaceae, and bismuth sulfide, which not only inhibits non-Enterobacteriaceae but also coliform, a subgroup in Enterobacteriaceae. Similar to XLD, the bismuth sulfide in the medium could be catalyzed into hydrogen sulfide, which could react with ferrous sulfate to generate black precipitation. The left picture showed you that a typical Salmonella colony on BS is black or green in color, usually with dark halo in surrounding agar. But it is possible that there is no dark halo. The colony itself and the agar surrounding it often have metallic sheen as shown in the right picture. TSA stands for tryptic soil agar. It is non-selective. This medium contains digests of casein and soybean meal that could supply nitrogen source in the form of amino acids and polypeptides. Therefore, this medium is highly nutritious and is good for growing a wide variety of fastidious and non-fastidious microorganisms. Colonies on selective and differential agar plates are grown at a stressful environment and likely they do not show their typical physiological reaction. Therefore, we do not conduct confirmation tests directly from colonies on selective and differential agar plates. Instead, we allow them to grow on a nutritious and non-selective agar plates first. A typical Salmonella colony on TSA is creamy, round, and smooth with slight rise in the center. TSI stands for triple sugar ion. It is a slant agar used for biochemical tests. It is called slant agar because, as shown in the picture, this agar contains a slant and a butt. Reactions that happen on the slant are aerobic, while reactions at the butt are anaerobic. This medium also contains 3 types of sugar: glucose, lactose, and sucrose in the proportion of 1 to 10 to 10. It has peptin as a nitrogen source, which is a secondary nutrient. On the slant, Salmonella first aerobically digests glucose, leaving the medium at neutral pH. After consuming all the glucose, Salmonella start to aerobically break down peptin, turning which into alkali molecules. The pH indicator phenol red will change to pink reddish color. In the butt, the Salmonella will ferment glucose to produce acids. Because the amount of glucose is very limited in this medium, the color change at the butt will not be extended to the area close to the slant. Whereas microorganisms that could ferment either one of the other 2 sugars would profusely produce acids. These acids could diffuse to the slant area, resulting in not only the butt but also the slant to turn yellow. Gas may be produced during the fermentation. Similar to the reaction in XLD, the sodium thiosulfate and ferric sulfate in the medium could result in black precipitation when Salmonella is present. As you can see, the positive tube for Salmonella shows red slant, yellow butt, and black precipitation. Sometimes the bacteria produce large amount of black precipitation that will mask the yellow butt. One thing you can do is to observe the tube against the light, and you might see some yellowness shining out from the black precipitation. You might see some crack in the agar or the entire agar get pushed up due to the gas production. LIA stands for lysine ion agar. It is also a slant agar used for biochemical tests. The primary nutrient, glucose, is scarce in this medium. However, the L-lysine, which could be used as the secondary nutrient, is generous. When Salmonella is inoculated, the reactions on the slant are the same as those happen on the slant of TSI. Therefore, the slant is alkaline, which allows the pH indicator from crystal purple to stay purple. In the butt, Salmonella first ferment glucose, making the agar acidic and show yellow color. Afterwards, Salmonella starts to use lysine as an energy source by catalyzing lysine into basic molecule through its lysine decarboxylase. It has to be pointed out that this reaction only happens anaerobically. Because the amount of lysine in this medium is way more than the glucose, the alkali generated could neutralize the acid, and the excessive alkali could make the agar basic and show purple color. Similarly, the sodium thiosulfate and ferric ammonium citrate in the medium could result in black precipitation when Salmonella is present. Positive tube for Salmonella shows purple slant, purple butt, and black precipitation. Similarly, sometimes the bacteria produce large amount of black precipitation that will mask the purple bud as shown in this picture. You can use the same method to see the color. You might see some crack in the agar or the entire agar get pushed up due to the gas production as well. To record the result of slant agar, a specific annotation method will be followed. The general format is to use a slash to separate the reaction on the slant and the reaction at the bud. Before the slash is the slant reaction, which could be either K indicating alkaline or A indicating acidic. After the slash is the bud reaction, which could either be K or A as well. Any other reactions such as precipitation written as plus or gas production written as G is annotated in the superscript. For example, the LIA tube in this picture showed purple or alkaline slant and yellow or acidic bud and no gas production or precipitation. Therefore, it is annotated as K/A. In the second example, this TSI tube showed both yellow or acidic on the slant and at the bud, and you can see cracks in the agar indicating gas production. This tube should be annotated as A/A superscript G. Similar to rapid detection, the serological test also detects the antigens presented on the surface of Salmonella. However, this reagent includes anti-Salmonella antibody against O antigen A2I as well as the VI antigen on the capsule. When Salmonella is present, the antigen will conjugate with the antibody resulting in agglutination as shown by the left reaction in the picture. If Salmonella is absent, there will only be a cream-colored smear as shown in the picture on the right.